All right, welcome to part 3 and the final segment of chapter 20. And we're going to discuss the rest of the chapter about DNA tools and biotechnology. This time we're going to focus on whole organism cloning and different methods related to that. All right, so cloning whole organisms. This is a controversial subject. So whole organisms have been cloned in the past, and it's important to know that stem cells are at the center of it all. And this whole process can be useful for use in basic research and other more practical applications. So organismal cloning is ultimately going to produce one or more organisms that should be genetically identical to the parent that donated the single cell. And that single cell is going to be a stem cell. And stem cells are considered what's called totipotent. Totipotent cells can generate a complete whole new organism. That cell can differentiate into any cell type with the right conditions. Cloning animals by nuclear transplantation was really where it all started. Let's discuss nuclear transplantation. So ultimately in nuclear transplantation, the nucleus of an unfertilized egg or cell is going to be replaced with the nucleus of a differentiated cell or less differentiated cell. So what this data showed that a more differentiated cell is less likely going to develop into a whole organism than a less differentiated cell. So these are data from this group down here. So let's take a look at what happens. So if we start out with a frog egg, which has been enucleated. So here UV radiation is applied to the frog egg, which destroys the nucleus. And so now we have this cell without a nucleus. This is going to serve as the donor cell. Okay, so the donor cell is going to receive a nucleus from a different type of cell. So here, what the group did was compare first a frog embryo with a less differentiated cell. So taking the nucleus from the less differentiated cell and injecting it into the enucleated cell, which is going to be this one right here. So that's going to be the recipient. And now we have the donor. Okay, so the donor nucleus is going to basically be injected into the recipient cell. And then they allowed it to grow and start to undergo development. And most of the tadpoles, or most of the frog embryos had developed into tadpoles. So it was pretty promising. As opposed, and this is as opposed to a fully differentiated cell. So this is where they took an intestinal cell, which was fully differentiated, and it's an intestinal cell, and they took the nucleus from that and then put it into the recipient, and then ultimately most of them stopped developing before the tadpole stage. So these data from this group indicate that a less differentiated cell is more likely going to undergo full development and to be able to be used to develop into a full organism than a cell that's already been differentiated into a certain cell. So cloning of a mammal was actually done by the method that we just described, nuclear transfer. In 1997, Scottish researchers cloned a sheep, and the clone was named Dolly, and this was done by nuclear transplantation from a differentiated mammary cell. So cloning has been developed in many mammals, including mice, cows, cats, horses, mules, pigs, and dogs, with mixed results. Unfortunately, Dolly did not live very long, and there are many reasons why this may be the case. And it may be due to epigenetic differences between the cloned animals and the type of animal they were cloned from. So only a small percentage of these cloned embryos have developed normally to birth, especially in the eukaryotic types of organisms. And it's mainly because the cloned animals exhibit defects, and it's because of epigenetic differences. And, what is epigenetics? So epigenetic changes must be reversed in the donor before the cloning occurs. But these types of epigenetics include acetylation of histones. So sometimes acetylation or deacetylation of histones are going to be different, and this is a method in which it can control gene expression. So acetylation of histones, you want to remove the acetylation because that affects the development, and basically these are changes at the chromosomal level that occur during development. So if they've already been acetylated, then that means they're genes that are silenced, and these could be genes that would be needed for a full proper development of the organism. Same thing with methylation. So methylation can silence genes as well. Now, stem cells of animals can ultimately reproduce indefinitely, and under certain conditions can differentiate into more than one type of cell. So if you look at this, a stem cell can undergo cell division and result in another stem cell and a progenitor cell. So the stem cell is going to stay a stem cell. However, the progenitor cell will eventually differentiate into different cell types. So you can have progenitor cells that are specific or will differentiate into fat cells or bone cells or white blood cells or red blood cells or any other cell type. So embryonic stem cells versus adult stem cells. So there are differences between the two. So embryonic stem cells are different from adult stem cells in that, first, adult stem cells can generate a limited number of cell types. So if we pull stem cells from the bone marrow and then culture them, they'll only develop into blood cells. And here we see red blood cells and white blood cells. But embryonic stem cells, if you pull stem cells from embryonic tissue and then culture them, they could potentially become blood cells or nerve cells or liver cells. This makes them pluripotent. Okay, so pluripotent means that they can differentiate into different cell types. Now, embryonic, and a little bit more about embryonic stem cells. So the main aim of cloning embryonic stem cells is to produce cells in order to treat disease, right? So this is really the good way of using embryonic stem cells rather than trying to clone whole humans. However, there are techniques that are being currently optimized to clone human embryos. Now, the main point, again, of using the embryonic stem cells were obtained only from the embryos donated by patients undergoing infertility treatments. Now, the process thus is called a therapeutic cloning. So this is the good, the more, I guess, accepted reason for using embryonic stem cells. Now, there are, of course, opinions that vary about the morality of therapeutic cloning, even if it's meant to for the good. So let's look at induced pluripotent stem cells. So these are a little bit different from basic embryonic stem cells. So retroviruses can induce extra copies of four stem cell master regulatory genes to produce induced pluripotent stem cells. Human induced pluripotent stem cells have been developed from individuals with type 1 diabetes, Parkinson's disease, Huntington's, Down syndrome, and many other diseases. They can be used in regenerative medicine. So this is a good thing about the induced pluripotent stem cells. All right, so let's look at induced pluripotent stem cells. Remember, they have been developed from several different types of patients with different diseases. So what this is, and these are data from this group down below in the citation. What this is taking progenitor cells, and basically these progenitor cells originated from a stem cell, but using the progenitor cells and then inducing them to produce the stem cell master regulatory genes. And these are going to be basically introduced by viral vectors, so that you have here, this is an example of a skin fibroblast cell. This is something that's differentiated. Well, you can basically convert it into an induced pluripotent stem cell. So you can take any type of already pre-differentiated cell and then convert it to a pluripotent stem cell. So this eliminates the need to use embryos. Now let's talk about the practical applications of these types of biotechnology, or any of the types of biotechnology discussed in the segment 1 and 2. So some practical applications, these have has affected us throughout the years. So biotechnology is really the manipulation of organisms or their components to make products that are useful. And it could be medical or agricultural or environmental ways. So some examples of DNA technology that's used in genetic engineering include those in medicine and forensics, and providing or obtaining genetic profiles, and environmental cleanup, and then of course genetically modified fruits and vegetables that are made to be able to withstand harsher conditions and or produce more nutritious fruits. So in terms of pharmaceutical products, so advances in this DNA technology and genetic research in general have been important in the development of useful drugs to treat diseases. One of them including the synthesis of small molecules for use as drugs. So you can manipulate organisms to synthesize these small molecules or potential pharmaceuticals. Protein production. So proteins like insulin are used to treat diseases. You can manipulate organisms to produce excess amounts of this protein, which make it easier, and you can produce high amounts of it at one time. And protein production by farm animals, right? So you can have farming animals, and we'll talk about that in just a second, and induce animals to actually produce these and extract them out of, as milk. So, of course, then the ability to do that is based on producing transgenic animals. And these are animals that are going to be expressing genes or transgenes, or genes from other organisms, other species. An example would be farm animals. So farm in quotations, PHARM, it implies that these animals are producing pharmaceuticals in their farm animals. So, of course, just a reminder, a transgene is a gene that's transferred from one organism to another. Transgenic animals can express this gene, which will ultimately result in the production of the gene product. Transgenic animals can be considered, or these farm animals are pharmaceutical factories. So they can be made to produce high amounts of the product of a particular gene. So here we can see that some farm animals can be engineered to produce or secrete a product within their milk. So all you have to do is collect the milk. It doesn't hurt the animals. You collect the milk and you have the protein product within the milk. You have to isolate it simply, and you can produce high amounts of it. In terms of forensic evidence and genetic profiles. Okay, so genetic profile is going to be basically, identifying individual variation between individuals. So this is, in forensics, the use of what's called short tandem repeats, or STRs. Okay, so what short tandem repeats are repeated DNA sequences, or very small sequences that could be 4 to 6 bases in length, and number of repeats. So each individual can have different numbers of repeats at particular loci within the genome. So STRs, again, they're variations in the number of repeats. They're analyzed old-fashionedly by PCR and gel electrophoresis. Now, there's other instruments that perform gel electrophoresis in a different manner, which makes it a little bit easier than what you saw in segment 1. But this allows scientists or forensic scientists to exclude someone from a crime by identifying the genetic profile, comparing those profiles from individuals might have left a sample at a crime scene, some sort of tissue or blood, and be able to include or identify an individual that might have been present, determine paternity, and identify victims of mass disasters. One key example, or really an example of excluding someone from a crime, unfortunately, this individual did spend some time in prison. Earl Washington had spent 17 years in prison for a crime. And this was before STR analysis was developed and used in defense, or be able to be allowed in the courtroom as a defense or prosecution for that matter. But once it was developed, it was reanalyzed. So this is part of what's called the Innocence Project, and this is a way to reanalyze samples now that we have better technology to exclude those or identify or confirm an individual that might have been committed a crime. So if we take a look at these data right here and how STR analysis can be used, there's actually 13 STRs that are used in forensic science, but this just shows an example of 3 of them. So we have different sources here. So this, he was accused of rape, and we have the semen, or the STR profile of semen that was found on the victim. Now this was again reanalyzed. We have the sample. Now it was analyzed, and the number of repeats at marker number 1 for the semen on the victim. So this is the source of information. So the semen on the victim could be, or is most likely, the individual that committed the crime. So 17 and 19. So remember, there are 2 chromosomes on each diploid individual. So each chromosome can have, or again, they could be heterozygous. They can have different number of repeats. So the semen had 17 and 19 STR1, 13 and 16 STR2, and 12 were the same. They were homozygous for STR3. Well, Earl Washington had a different profile. So after analyzing Earl Washington's DNA, it was determined that he did not match the semen on the victim. And there was another suspect, Kenneth Tinsley, which was then convicted. So Kenneth Tinsley did eventually plead guilty to the murder and the rape and murder of this individual. So you can see that Kenneth Tinsley, the repeats on his profile match that of the semen on the victim. And this is how these markers are used to determine paternity and to identify an individual that may have committed a crime, and then identify individuals that may have been victims of mass disasters. Let's look at genetic engineering. Of course, you've heard of this in genetically modified organisms, but one good way to use it is to clean up the environment. So part of this might be a wastewater treatment, right? So you want to treat the water for microorganisms. So if you can modify the metabolism of certain organisms, you can extract minerals from the environment, and you can degrade toxic waste materials. So you can modify the metabolism of these microorganisms so that they can extract the minerals from the environment or the wastewater, or degrade toxic waste materials that may be in water or may have been spilled, like an oil spill. They have the potential to degrade hydrocarbons and other harmful compounds. In terms of agricultural applications, you might want to genetically engineer certain crops to be able to withstand certain conditions. For instance, you might want to improve the productivity or the yield of plants. So remember the difference between C4 and C3 photosynthesis? So those organisms that undergo C3 photosynthesis can be affected more by conditions where there's less water, right? So if there is a drought. C4 plants, and remember those are ones that can live in really hot and dry climates. So if you can engineer plants to undergo C4 photosynthesis rather than C3 photosynthesis, you can um, uh, avoid that type of situation. You can genetically modifying plants, you just basically speed up the selective breeding process because normal breeding takes a long time. So you would have to physically cross them and then wait for the offspring and then pick out the offspring that have your desired characteristics and then mate those and then continually go through that process over several generations. So genetic engineering can skip several generations and really be engineered within one generation. You can engineer plants to have herbicide or pesticide resistance so that you can spray them and kill plants or kill animals or insects that may affect crops and not have the herbicide or the pesticide resistance or the herbicide or pesticide affect its growth or crop yield. You can engineer plants to be resistant to salinity. So this is kind of related to the drought situation. So when there are environments where, let's say, the less water can increase the salinity or the salt concentration within the environment, that could affect crop yield as well. So if you can genetically engineer them to resist high salinity or high salt conditions, then you would be able to circumvent that and still have a good yield of your crops. And then also you can engineer crops to have better nutritional values. So going back to, let's say, rice. Rice has just starch. It doesn't really have many vitamins in it. So if you can engineer rice to produce vitamins like maybe vitamin B or iron, then it would be more nutritional, and that's a benefit to those countries that depend on rice as a sole food source. Now, in terms of safety and ethical questions raised, you might be already thinking about this, right? So, what is the possibility of producing hazardous new pathogens, right? So, bioterrorism, that's a concern, a valid concern. Possible hazards centers on the genetically modified organisms, typically as food. So what are some questions might be raised in terms of the environmental consequences of genetically modified organisms? What about the consequences on humans? For instance, those individuals that might have allergic reactions to some component that's within a genetically modified organism. So these are some important questions that I want to kind of conclude with, and maybe you want to think about these. So under what circumstances, if any, should genomes of human germlines be altered? Okay, so is this a moral problem? So should we, scientists, be playing and altering, basically playing around with natural selection? Would alterations lead to deliberate efforts to control the genetic makeup of human populations? Right, so this is that area called eugenics, right? So if you're producing certain traits within human populations on purpose. In terms of gene therapy, right? So there are some questions that are raised in terms of technical issues. How can the activity of a transferred gene be controlled to make appropriate amounts of the gene product at the right time and place? You do not want to produce too much of the gene product. You want to make sure that it's produced at the right time, in the right location. How can we be sure that the insertion of a therapeutic gene does not harm other cellular functions? So if you're producing a protein that should not be, maybe isn't normally present in your cells, or maybe not normally present in such high amounts, is it going to affect other things that are happening in the cell? So basically, as we learn more about these controls, or regulatory controls, and the interactions between genes, we may be able to answer these questions. All right, thank you for listening.